Hello everybody and welcome back to the lectures. So we have been discussing about the protein sequencing and that was in module 6. So if the title of the module 6 was protein sequencing and solid phase peptide synthesis. We have completed the protein sequencing and I think it is a good idea to break this module into two. So today we will start with module 7 and that is regarding the synthesis of the peptide, including the solid phase peptide synthesis. So the module 7 will be about chemical synthesis of peptides and how the peptides could be used as therapeutics. So if you have two amino acids, how would a peptide bond be synthesized? Let's take the example of maybe an alanine which has the CH3 group. This is your NH2, this is your carboxylic acid. Of course, it exists in zwitterionic form, but I am writing the neutral one. And you take another amino acid, let us say phenylalanine. Acid and the NH2. So if you condense them together, it will release one molecule of water. It's basically reaction between this carbonyl and this amine, acid base reaction, eliminating one molecule of water. But this reaction does not happen easily. If you just mix the two amino acids together, they simply do not react. Amino acids are not very reactive. So, you need certain reagents to make them reactive. So, you need a reagent for this reaction. I will talk about the reagent just a little bit later. So, if you fuse these two amino acids, you can expect to get a peptide bond. This is alanine, this is phenylalanine, so you get a dipeptide. Dipeptide means it is a single peptide bond which has two amino acids, and then you have a free carboxylic acid group. Now again, if you add another amino acid to this, let us say valine, acid group, amine, then again you can expect a condensation here. Phenylalanine, this acid would become a peptide bond and you will have valine with another free carboxylic acid group. I know there are problems here, I am not talking about those right now. If you go on, so you have three amino acids linked together now. If you go on like this, then you can expect to synthesize a short sequence of peptide. Short sequence of amino acids which we call peptides. So this is a chemical synthesis, and nowadays we can synthesize 30, 40, 50, 60 to 70 amino acids long peptides can be synthesized in the laboratory, and I will tell you later that most of those long chain synthesis we usually do in solid phase, that is called the solid phase peptide synthesis. I will come to that later. So that is how, this is a rough scheme how to synthesize the peptides in the laboratory. So, the question is, why do you need to synthesize the peptides? What is the need for the short peptide synthesis? Okay, before that, so your target is to synthesize a peptide bond, and that occurs by the fusion or by condensation of an carboxylic acid group and the amine group. Now, if you look at the structure of all the amino acids that are present, you can see a lot of variations in their functional groups or in their structures. Of course, mainly in the side chain here. R group varies a lot, and there are plenty of different functional groups present in different amino acids. Peptide synthesis is very different compared to the DNA synthesis we have done. For DNA, the nucleobases itself, they do not have many functional groups, but those a lot of variations exist in amino acids. There are basic side chains, there are acidic side chains, here also amide bonds are there, sulfur groups are present which are reactive, the alcohols, OH functional groups, and so on. So, whenever you are doing a peptide synthesis, you also have to take care of these functional groups of the side chains. So, that is one chemistry aspect in the perspective of organic synthesis, that whenever your aim is to synthesize only the peptide bond, you have to be very careful about the side chains, so that they do not react or they do not interfere into your synthesis. But on the other hand, this also gives you immense opportunity to vary the modifications, to vary the structure of peptides. You can use these functional groups to make new kinds of peptides in a branched way or whatever way you like. You have the way out to make mold, to modify your peptides, so that you can synthesize new molecular probes or new molecules. So, the question is, why do you need to synthesize the smaller version of peptides, the shorter sequences of the amino acids? They are not proteins. So, the first importance of it, the first one is that, of course, if you want to study the biological property of a protein, then you can use, instead of the whole protein, you can use the short sequence of the area of interest of the protein, and to use it and use that to study the or to understand the biological properties or the biological functions of the protein. For example, you want to know how the active site of a protein is bound to its substrate, how it is bound to its cofactors, what is the chemistry behind the catalysis of the within the active site of the protein and the substrates that are coming in. So, in order to know that, many times if you take the whole protein, the things becomes very complicated, the chemistry becomes very complicated, the analysis of the data becomes very complicated. So, if you use, instead of the whole protein, if you use the peptide which constitutes the active site of the protein, then you can use that only the short sequence of it, and to study the biology, to study the binding properties or the other physiological properties that you want to know. And data becomes very easy to analyze, because you have a short sequence. It's things gets easier for handling purpose also. So, that is one way, one aspect or one importance of the smaller peptides, that to understand certain biological phenomena for example how the active site, this is just one example of it, there can be plenty of other biological functions, how active site works, or how is it bound to its substrate or coenzymes. All these are very essential to understand, of course, that how the catalysis works, what are the properties of the binding between the substrate and the peptide and the protein active site, which kind of coenzymes are involved, how the coenzymes are stacked onto the pocket of the protein. So, in order to understand that, you can use these things. And another very important aspect of using the short peptides is that you can do crystallography very easily, I mean rather easily compared to the whole protein. Whole protein is very tough to crystallize. So, if you want to see actually the structure of the active site substrate combination in a frozen form, then you have to go to the crystallography, you have to form the crystals, and then the short sequence of the peptides helps a lot in getting a good crystals. So, that is one aspect of it. Second is there are many short peptide sequences that we know of nowadays that have therapeutic purpose, and that can be used to cure as well as to take other molecules into the different targeted sites of the diseased cells. So, the second aspect is therapeutics. Not only therapeutics, if you want to do a certain imaging, you want to take the fluorescence molecule or other markers to certain part of in the cell, then sometimes peptides also help you to take those molecules to the specific positions. So, there are many peptides nowadays which are known and which have tremendous applications in drug discovery and drug delivery. Therapeutic, drug discovery, drug delivery. For example, CRECA, CREKA, this is a peptide sequence, and of course you can see it is, do you remember the alphabets which amino acids they represent? You have to look at the here you can find out actually, here all these alphabets are written, symbols are written. So, it is a good practice to know or to remember all the alphabets which represent the amino acids, because that is how usually a protein sequence is written. Protein is too long, so you cannot write the individual amino acids name, it will take too long time. So, it is represented usually, if you see a protein structure, protein primary sequence somewhere, you will see these kinds of alphabets are written. Of course, they represent the amino acids. So, CRECA 1, 2, 3, 4, 5 is a pentapeptide. What is the sequence? C for cysteine, R is arginine, E is glutamic acid, E is glutamic acid, K is lysine, and A is alanine. So, this is the sequence of this peptide, CRECA peptide, and this peptide has found enormous importance. This is actually a tumor targeting peptide. You can write TOMOR or TOMOUR. Tumor targeting peptide. That means this peptide can go and adhere to a tumor cell, because tumor cells have receptors to receive where tumor cells have certain receptors where this peptide can go and bind. So, the normal cells does not have them, specifically tumor cells have them. So, if you have this peptide and if you try to send it, then this peptide will specifically go and be bound to tumor cells. So, this will be, this peptide binds or adheres to the tumor cells, because the tumor cells have the receptors to dock this pentapeptide. And of course, you can find now that how important this would be. It is usually very difficult to differentiate between a normal cell and a tumor cell. So, therefore, whenever you try to send a drug to cure the tumor cells or to kill the tumor cells, they also kill the normal cells. So, of course, our aim all the time has been how to make a difference, how to take the drug only to the tumor cells which will not affect the normal cells, and that has been still going on. I mean, there are plenty of, that is the major problem associated with tumors or associated with cancer, that all the drugs, all the therapies, all the chemotherapies, all the radiations that are there, all those treatment techniques that are available, they are also harmful, very harmful in fact, for the normal cells, and that is a reason that the tumor cells cannot be selectively killed. So, if you, as I was saying, if you want to take a drug to specifically to tumor cells, there are not many ways. It also goes to the normal cells and kill the normal cells. So, now if this peptide only goes or binds to the tumor cell, then the advantage is, if you conjugate a drug here or if you attach a drug along with this, then what will happen? The drug along with the peptide can now go specifically to the tumor cells. So, that is called the targeted drug delivery. So, such kind of short peptide sequence, because of its tumor targeting property, can be very useful in therapeutics. Not only therapeutics, for our academic purpose, whenever you want to understand something, you want to study a phenomena, you want to study a physical property or biological property that is going on into the tumor cells, and you want to take certain other molecule, for example, an inhibitor or a fluorescence marker molecule to it, you can always attach that here, and this will go specifically to the tumor cells to give you the reports. Similarly, another peptide which is a tripeptide, RGD, is a tripeptide what is the sequence? R is arginine, G is glycine, and D is aspartic acid. So, it is a tripeptide, but often this exists in a cyclic form. RGD peptides often exist in a cyclic form, and it can have a polymeric structure also, polymeric cyclic, maybe a three member of RGD can be present. So, this peptide RGD or another one called NGR is also a tripeptide which has the sequence of N is asparagine. I am writing the full form here to differentiate between aspartic acid. G is your glycine and R is arginine. So, very close structures actually, very close sequences. This has arginine, this has arginine, this has glycine, this has glycine, only aspartic acid and asparagine are different. These two peptides are known as tumor homing peptide. Almost similar to CRECA, these peptides will also have receptors in tumor cells. So, they can go and specifically bind to the tumor cells. Tumor homing peptide means they can go and stay with the tumor cells. So, again similarly these peptide sequences can also be applied or can also be very useful in understanding the biological functions that is going on in the tumor cells, as well as to carry drug molecules to the tumor cells and do a treatment. These kinds of peptides I have talked, there are other examples also. These are the most well studied peptides that are used to target the tumor cells. Now, another kind of peptide sequences, mostly the charged peptides, peptide which has multiple positive charge. For example, polyarginine sequence, or polylysine sequences. Arginine and glycine both have amine as side chains. So, obviously they will exist as in the zwitterionic form, they will exist as NH3 plus that has positive charge. If you have a polymer of arginine, if you have polymer of lysine, they will impose lot of positive charge in your peptide sequence, and those charged polar peptides are very good in penetrating the cells. So, they are known as cell penetrating peptides. In short, they are called CPP. So, those kinds of molecules with high positive charge and polar side chains can cross the cell barriers, can penetrate into the cells, and that is very important because our cell walls are pretty protective, pretty strong. Usually our cells do not allow any foreign bodies or any foreign entities to cross into its membrane, because they are outsiders and they can harm the cells. So, cells are very protective not to allow any foreign, any unknown bodies or any unknown molecules to penetrate into the cell, and that is where most of the drugs that you use do not get through the cells. So, only a certain percentage of the drugs that we actually use can penetrate into the cells and can do its functions. The rest of the drug are actually eluted out or will be coming out, excreted out of your body, and they do not go into the cell. So, in order to increase the bioavailability, in order to increase the amount of your drug inside the cells, you need something that will help you to cross into the cell barriers, and that is where these peptides are for, CPP. So, if you attach drugs here, if you take other molecules that you want to take into the cell, this is a good idea that you can combine the cell penetrating peptide along with those molecules, and then it can together they can get into the cells. And this thing, cell penetration, becomes much more harder when you think of brain cells. So, our brain cells, brain is the most important part of our body. So, brain cells are overprotective not to take any molecules from outside. It is very hard to send drug molecules or medicine into the brain cells. They simply get rejected. So, there are blood brain barrier peptides. blood brain barrier. In short, they are called BBB. Peptides. So those peptides can cross the blood cells, and therefore obviously they have really huge importance in biology, in drug delivery, in pharmaceuticals. So there are many again there are many sequences that are known nowadays. Some of them are very active, some of them are not that active. So one of these is a TAT peptide. TAT is actually a whole sequence of a protein, and particular part of it, the sequence 47 to 57, this 10-mer sequence I think in the whole TAT sequence. So I call it TAT peptide. That is a blood brain barrier peptide. It can cross the blood brain. So blood brain barrier peptides are peptides that can cross or penetrate the brain cells or blood brain, that can actually penetrate the brain cells. So this peptide, a certain sequence of the TAT peptide is, I will write down the sequence of it: Y, G, R, K, K, R, R, Q, R, R, R. So you can see here a lot of R, arginine. So as I was saying that for any cell penetration you need more positive charge, positively charged amino acids. So therefore the number of arginine should be more, polar side chains. What is the full form? Y is your tyrosine, glycine, arginine, K is lysine. So it is full of lysine and arginine. Again K is lysine, arginine, arginine. Q is your glutamine, GLN, glutamine, arginine, arginine, arginine. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So it is a 11-mer peptide sequence that will allow you to cross the blood brain cells. So these are some of the important reasons why we need to synthesize peptides that I have talked about. There are many others. So now coming back to the synthesis. So peptide synthesis. As I have said, if you want to synthesize a long sequence of peptides, then you go for solid phase peptide synthesis. But the short sequences of the peptides, for example 3, 4, 5 up to 10 even, we can synthesize in solution phase itself. So let us start with the solution phase peptide synthesis first. So this should be solution phase peptide synthesis for the time being. If you do it in solution, if you take 2 amino acids again, let us say you have R1 here and R2. You add the reagent. What is your product going to be? In solution, you have both the molecules. So, obviously there will be condensation here. R2, this is 1. Is there a second possibility? So, in this case, the acid of R1 is reacting with the amine of R2. What can happen? The other way around. The acid of the R2 can react with the amine of the R1. So, in this case, you will have R2 R1. This is a different molecule, this is a different molecule. So, we have already two possibilities. Is there any other possibility? Of course, there are. They can go self condensation. So, one molecule of R1 can react with the other molecule of R1 itself to give you one peptide. So, R1, so it is a homodimer. So, both are R1. This is also possible. Similarly, for the R2. Both are true. So, you have at least 4 possibilities. If you do a solution phase reaction between 2 amino acids, I am just talking about 2 amino acids to synthesize a dipeptide, and all 4 will be formed in your solution. Now, you have to separate them out and then of course, you can see that separation will be a very complicated factor, because all of these are dipeptide. So, lengthwise they are same, they have same number of free acid and same number of free amine groups. So, their polarity will be roughly the same. If the side chains are not very different, then the separation would be very difficult actually, if you have that many possibilities of product formations. So, what is the way out? If you synthesize, if you want to do it in solution phase itself, just a dipeptide first, what is the way out? Of course, the way out is to do selective protection. For example, if I want to have only R1, R2, this molecule, then I have to inactivate this amine and I have to inactivate this acid. So, selective protection of the amine group here, selective protection of the acid group here, and then mix them together with the reagent, you can expect to get your desired product. So, first, how to do the amine protection? So, if you have so this is our desired product. Therefore, what you have to do is keep the acid group free and protect the amine. I am terming it as P1. So, this is amine protection. That you have to do for the first amino acid. What about R2? For R2, you need the amine group here because there is acid free. Here an amine should be free, and your carboxylate should be protected so that it does not react. So, this should be carboxyl acid protection. So, two different amino acids should be protected selectively. And now, let us say this is your A and I term this as B. Then if you now mix A and B in presence of the reagent, then you can expect a AB peptide. And of course, in this A, there should be acid protection, there would be the base protection, and then D protection. So, these are the additional synthetic steps that you have to use for the D protection of this first and then D protection of this. Two different reagents obviously would be required. Then you will get back your desired peptide. This is for dipeptide. Now, if you want to go for the longer chain peptide sequences, then what are the possibilities to do them? You have R1 acid NH2. So, first step, what you can do, of course, you have to protect the amine. Leave this on. This is the first, and then you add to it with the second amino acid, which will have a free amine group and a protected acid, as we have done before. Then you get NH R2 COO P2. Now, what you have to do, if you want to carry on the synthesis, you have to make this acid free. So, therefore, D protection of P2, selective D protection of P2. So, one way of it. This can still be protected R2. This will give you a free carboxylic acid. Now, if you want to carry on, bring in one R3. What you need is a free amine here. P2 protected carboxylic acid. So, you can see after the first one, all the subsequent amino acids that you will be needing does not require protection of the amine group. If you follow this kind of protocol, it only requires the protection of the carboxylate, and it can go on like this: NH P1 R2, R3, P2, then again the protection of P2. It will give you another carboxylic free and you can move on like that. So, this is one way of it. It can be done other way around also, where you start with the protected carboxylic acid and do the reaction on the amine part. So, this I think the other way around, where we will have protection on the amine, not on the acid, and that is mostly used. We use it for, especially for solid phase peptide synthesis, we use the protection of the amine group, amine side chains, that we will see. So, this is for the protection of the acid group here, and before I have talked that we can protect the amine group also. So, both protections sometimes would be required. Now, the question is, what kind of protecting groups you will be using? So, protection or protecting groups. Let us start with the amine. How to selectively protect the amine group here? There are many reagents. The reagents which we use most are two of them. One is called BOC, another is called FMOC. These two are the different protecting groups or protecting group chemistry that we use for amine protections of the amino acids. So, first is with the BOC. BOC anhydride is the reagent. You can write BOC anhydride, or you can also write BOC2O, that represents BOC anhydride. Then you get R COOH NH BOC. That is the selective protections of the amine group. Now, how does it work? What is the structure of BOC? BOC is basically, this is BOC anhydride. Tertiary butoxy carbonyl anhydride. What is the structure? This O CO O, this tertiary butyl group linked to a oxygen here. It is basically a ester bond, acyl bond here, and then this is the anhydride part. How does it work? Obviously, now you can see the chemistry that is supposed to go on. This is electron deficient carbon. It is a nucleophilic center. Obviously, carboxylic acid won't react here. So, it will react, it will attack this, and it will come back, it will come here and go back there. Ultimately, it will cleave this bond, right. So, you will have NH, CO, O, this we call as BOC, and obviously it decreases the nucleophilicity of the nitrogen. So, the electron lone pair would be dispersed here. and that is the reason that this amine is not reactive anymore. This biochemistry is usually being used a lot when we do peptide synthesis in solution phase. Another is the FMOC chemistry. FMOC chemistry is mostly used when we do solid phase peptide synthesis. This is FMOC chloride. This is a fluorine moiety that is connected with the intervening CH2 group with a acyl chloride. So, again the same reaction, if you have R, acid chloride, obviously chloro would be out. NH, the rest of it we call FMOC. So, FMOC protection group is used to protect the amine group of the amino acid. FMOC chemistry is a little bit difficult to do in solution phase because the separation or the separation and dry up becomes a little bit difficult. So, that is why we prefer biochemistry for solution phase, FMOC chemistry for solid phase. So, that is about the amine protections. Now comes the acid protection. Usually, to get a good protection of the acids, we use ester formations because ester bonds are very strong bonds. So, we will synthesize ester. It can be done with the ethanol, with ethyl ester. That will make the acid pretty much unreactive, or you can use the benzyl ester, PHCH2OH, even the other ones, or COO CH2PH. These are the certain protecting groups of the acids. Now, of course, the deprotection chemistry I have not talked about. After the reaction is over, you need to deprotect them, right. So, deprotection for the esters is usually done, it is a little bit harsh condition actually, H2 palladium. So, that is why these are usually doing here also the same thing, same reagent H2 palladium or H2 platinum can be used to deprotect the acid groups. So, that is actually problematic if you have to do, if you want to synthesize the peptides in solution phase, every time you are bringing in a reaction, for example, here you have to deprotect the P2 here. So, hydrogen palladium again to deprotect the acid, then you have to go to the next step, again a deprotection with hydrogen palladium, that takes time, it is also a little bit harsh conditions. So, that is a disadvantage of doing the solution phase peptide chemistry. The BOC and the FMOC can be deprotected by acids, that I will show you when we do the solid phase peptide synthesis. Now, we have talked about the protections. Now remember, I was always talking about a reagent, that whenever you want to synthesize a dipeptide or you want to make a new peptide bond formation, you always need a reagent, because the amino acids are not very reactive. So another thing that I otherwise will forget to mention, I also said that the side chains, we have seen the pretty much different functional groups are present in the side chain of the amino acids. So whenever you want to do the peptide chemistry, you also have to protect the side chains accordingly, appropriately, and then deprotect it also. Sometimes it can be the amine groups has to be protected, the acid groups have to be protected, but you have to use different protection groups for those the side chain amino acids, side chain amines or the side chain acids compared to this. Otherwise, while deprotecting, those will be deprotected at every step also, that you do not want. Okay. So now the coupling reagent, the reagent that is used to couple the free carboxylic acid with the free amine are called the coupling reagents, and there are many different types of coupling reagents are present. Without the coupling reagents, peptide synthesis does not happen, because both the carboxylic group and the amine group in the amino acids are not very reactive. So you need to activate. These are some of the structures of the coupling reagents. There are many more. I will include one more here, DCC. So DCC is another one. DCC has the structure, it's carbodiimide, right? N double one C, double one N, cyclohexane. Dicyclohexane carbodiimide, that's a DCC. This is also a very famous coupling reagent. In fact, DCC was one of the first coupling reagent that was used for peptide coupling. So all these coupling reagents are mostly used to activate the carboxylic group. These carboxylate are usually targeted to be activated. So once you activate this, then the free amine can react. So you can see the complicated structure. This is BOP, which has a phosphorus atom here with a positive charge, and then fused with a 6-membered and a 5-membered ring, and the 5-membered ring is with the 3 nitrogen, right? So it's aromatic kind of compound. This is BOP. This is a HA2. HA2 has again this kind of structure, which makes with the electron deficient nitrogen. Here obviously there are charged species, all these are charged species, most of them. This here the charge separation is neutralized, here you have a positive charge, so you need a salt. PF6 minus is the stable salt. Here also this is HBTU. HBTU has again the fused structure with NNN triazine, it is a cyclic triazine actually, and then with this kind of you will see this is actually a derivative of urea that you can get as a side product, and then again PF6 minus and DCC. So next lecture we will see how the mechanism works, how the coupling reagents are involved in activating the carboxylic acid, and how the peptide bond formation actually works in presence of these coupling reagents. And then we will also move on to the solid phase peptide synthesis. Thank you.